I'm working on campus in the Fly Lab with Professor Norma Velasquez, another baseball teammate, Luke Bass, and then a couple other younger undergrads on campus. And right now I'm working to characterize the effects of long-term nicotine exposure on the adult Drosophila fruit fly. The nicotine and tobacco epidemic is still absolutely continue, with tobacco alone killing upwards of 8 to 10 million people a year from both direct and secondhand exposure. Not to mention the e-cigarette epidemic that is just growing. In terms of why we use fruit flies, there's a large amount of shared genome between humans and fruit flies that allows us to make certain comparisons, especially when looking at genetic differences and overall behavioral and survival trends. So for example, when you're using a mouse, that might be a little closer to a human genome and closer to what a human reaction would be. However, they have a longer lifespan. And so where these fruit flies come into play is we're able to run through multiple generations very quickly. We're able to expose these fruit flies to long-term exposure, but it might take a week or so to actually do that. So we can make these genetic and behavioral comparisons loosely while still doing them quickly. The first one we wanted to characterize was the survival data. So looking at if we were to put these fruit flies on varying levels of nicotine over time, how would that decrease how long they live? And so that's the first thing we checked. And then using that data, we were able to kind of choose a baseline number for how long and how much nicotine we want fruit flies to be exposed to before doing other tests. Using that number, we then did some behavioral tests, for example, on the olfactory sense of smell. And we also did some behavior tests in terms of how much they move. So like their fidget response, if they're just jumpy. We found that nicotine tends to increase how much females, flies in particular, just move around in their day-to-day activities. So that was really interesting. From there, we also wanted to look at a pattern of nicotine consumption, which, for example, in a human who's starting to use cigarettes, they would need more cigarettes over time to get that same feeling as that first time they used. So in fruit flies, we see a similar pattern where if they're given access to their own source of nicotine, they increase their consumption over time. And then finally, we're looking at how the effect of long-term exposure affects the dopamine neurons in the brain. And so we're doing some dissection, some immunostaining using some specific antibodies, and so we're looking at that right now currently. Early in March, I was able to, with the support of SAB, Lewis and Clark Student Advisory Board, I was able to go to Chicago and present at the National Fruit Fly Conference that takes place every year. And this one was really cool. It was a very large convention with over 1,000 presentations there from all over the world. There's undergraduate presentations, PhD presentations, professors doing their dissertation work, all sorts of really interesting stuff there. So it was a little more of an official large venue, but I was still able to get to know people in a casual way and get that experience presenting in a high-stakes environment. I took that experience and presented at a little more casual event called the Northwest Science Association and American Association for the Advancement of Science Pacific Division annual meeting up in Bellingham this past couple weeks, where I was able to present with a whole range of scientists, physicists, botanists, everything. And then I was able to walk away from there with the President's Award for my research and my presentation, which was a really cool experience. So being a D3 athlete, I think, has played a huge role in this process. One thing I know with my college commitment process was that a lot of schools kind of discourage students from pursuing sciences in general, much less research, where Lewis and Clark was definitely very excited to have people willing to pursue both athletics and research. So I think in terms of having the support of both my coaches and my teammates through this commitment has been really interesting, because the time commitment is very different than a class where you have an hour and a half twice a week, an hour 3 times a week, where you might need to go into the lab for multiple times a day. You might need a couple hours on the weekends. So just having the flexibility and support of those around me has been really huge in this process. For me, the next step is to finish up my thesis this month right before graduation, present that, hopefully all that goes well. And then we're going to look to finish up a couple experiments without me actually in the lab this summer, and hopefully get most of my results published this coming summer. And then hopefully next year I'll be attending either graduate school or medical school and continuing research in more of a clinical fashion.